Hello, everyone, and welcome to our 680th episode of People Behind the Science. Today, we are excited to reshare our interview with Dr. Gregory Petsko, originally published on June 15th of 2015. Listeners, Greg is a structural biologist and a biochemist by training, but his research interests have shifted to focus on understanding the biochemical bases of neurological diseases. He ultimately hopes to find cures for neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, and Lou Gehrig's disease. And over the course of his career, Greg has made important contributions to the fields of protein crystallography, biochemistry, biophysics, enzymology, and neuroscience. So listeners, get ready to hear from another one of our fantastic scientists in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Gregory Petsko. So welcome to our show, Greg. How are you?
I'm well, thanks. And yourself?
I'm doing quite well myself, and I'm thrilled to be here, as I said, with you and our listeners today. So listeners, just a brief introduction here. Greg is the Arthur J. Mahon Professor of Neurology and Neuroscience and Director of the Helen and Robert Appel Alzheimer's Disease Research Institute at Weill Cornell Medical College, as well as the Tauber Professor of Biochemistry and Chemistry Emeritus at Brandeis University. He received his PhD from the University of Oxford and worked at Wayne State University, MIT, and Brandeis University before joining the faculty at Cornell, where he is today. Now, Greg has received many awards and honors during his career, including the Pfizer Award in Enzyme Chemistry of the American Chemical Society and the Max Planck Prize. Greg is also a member of the National Academy of Sciences, the Institute of Medicine, the American Academy of Arts and Sciences, the American Philosophical Society, and he is the past president of the American Society for Biochemistry and Molecular Biology, as well as the current president of the International Union of Biochemistry and Molecular Biology. Now, Greg has also written a column on Science and Society that is available as a book entitled Gregory Petsko in Genome Biology, The First 10 Years. And listeners, Greg is here with us today to share his journey through life in science. So, Greg, we want to get to know you today, both as a scientist, but also as a person. So can you tell us first, what do you like to do when you're not doing science?
Well, sleeping would be good, but I seem to do less of that than I wish.
Right. Unfortunately, the case.
I can tell you, though, that writing about science and society and other things, because my column pretty much covered anything that came into my mind, including such bizarre topics as the Atkins diet and how scientists behave at cocktail parties, that was fun. I like writing for fun. It's much more interesting than writing fairly dull scientific papers. I should say, however, in the interest of full disclosure, that the columns guest written by my two dogs, Mink and Clifford, were much more popular than the ones I wrote myself.
Oh, wow. I'll have to check those out next. Fantastic. So we've heard a little bit more about what you like to do when you're not at work. Greg, tell us next about your science. How do you describe what you do to someone who's not familiar with your field?
Well, before I got into my current area of research, which is neurodegenerative diseases, it was actually not straightforward. And I used to talk about it in terms of locks and keys and trying to figure out how something fits together. That I was the kind of person who could tell you what a lock looked like if you wanted to know how a key worked. That was the way I used to discuss structural biology with people who were not scientists. But since I started working on human diseases, it's gotten a lot easier. I basically just say, I'm trying to cure Alzheimer's disease, to which people respond, boy, I hope you get there soon.
Right.
To which I respond, I get that a lot.
So are you close, would you say?
Me personally, who knows? The field in general is a lot closer than it was even a few years ago. So I was somewhat in despair about the prospects for finding a treatment for this disease in the immediate future as little as 6 or 7 years ago. But there have been enough developments of late that I'm really quite a bit more optimistic now.
Well, that's very exciting to hear. I know I myself am a researcher in Parkinson's disease on the neurorehabilitation side, and I get that question a lot with Parkinson's disease. You don't always have a great answer. Sort of, we're getting closer. We're always getting closer, but you can never pinpoint, within the next 3 years, within the next 2 years, which is, I think, what people want to hear a lot of the time.
Absolutely. Parkinson's is one of the 3 diseases we work on in my lab as well.
Well, fantastic. I'm excited to chat more about your research as we go through. But let's start off with a tidbit of motivation. Can you share with us, Greg, what is your very personal motivational quote or saying?
I changed my whole area of research when I was in my early 50s. And that meant starting afresh in a field I knew nothing about, being essentially a totally out of it bozo for years. Something, by the way, I was used to feeling at home. Mm-hmm. But I think what kept me going in part there was a line from Tennyson's poem Ulysses. Do you know this one? It goes something like this. Death closes all, but something ere the end, some work of noble note may yet be done. And I think that idea is what got me started in working on such a tough problem as Alzheimer's disease, Parkinson's disease, and Lou Gehrig's disease. And it's what keeps me going when we run up against the countless failures that you have in trying to find a treatment for a human disease. I don't know that we'll ever succeed, but I do think this is work of noble note. And I have to say that if I'm going to spend the last years of my scientific life doing something, I'd like it to be something of noble note.
Definitely. Well, I think you read that quite well. I like the dramatic reading there. And that is a fantastic passage for people to keep in their mind, particularly people who are in the areas of medical research and human disease. And I think it is one of the motivating factors that keep people moving forward, just kind of knowing what's out there, knowing what's to come in the future for ourselves and our fellow man and what we can do to try and stop it. So great quote there. Now, can you tell us a little bit about some of your role models? I think scientists get to work with so many inspirational and motivational people throughout their careers. Who are some of these people who had a big impact on you?
Well, I've had the enormous good fortune of having been mentored by people who were not just terrific scientists, but who were fantastic human beings. And several of them come to mind immediately. One is Bob Langridge, who was a professor of biology at Princeton University, with whom I did undergraduate research. Another was a chemist at Princeton named Bill Horrocks. The two of them were tremendously important to me. Then my graduate student mentor, Sir David Phillips in England, who was both one of the world's greatest scientists, but also someone who became essentially like a second father to me. And later, when I got older, a tremendously close personal friend. My postdoctoral supervisor, Pierre Duzou in Paris, a man just absolutely wonderful in terms of his ability to enjoy life and who taught me so much about living as well as a good bit about science. These have been people who had, in my formative years, a tremendous influence on the way I did my science, but also the way I tried to live my life.
Well, I love these different examples of lessons learned from your different mentors throughout the years. Are there certain traits that you feel like you really picked up on and brought into your own style of doing research?
Well, I wouldn't be so bold as to say that I've succeeded in bringing them in, but I've certainly tried. I think what every one of those people had in common was the fact that they paid as much attention to the human side of things as they did to the science side of things. They cared about the way their laboratories functioned as people environments. They were people who were concerned about you, not just while you worked for them, but for decades afterwards. They were great human beings as well as great scientists, and it gave me the motivation to try to not neglect the human side of affairs as I pursued my scientific career.
I think you brought up a really great point, and this is one of the reasons why we love having this podcast as an opportunity to showcase the human side of science, to show that researchers aren't these one-dimensional people going into the lab every day and, that's their life. There's so much outside of it, and people are so multidimensional, and I love that you described having the opportunities to be in these laboratories where the environment, the human side, was a really big component as well.
So after I became a functioning independent scientist, I think there were two people who had tremendous impact on my career. One was Alex Rich, who's thankfully still with us, a professor at MIT. And the other, unfortunately no longer with us, is Dan Koshland, who was professor at Berkeley. When senior scientists of their stature take an interest in you when you're starting your career, it can have a tremendous effect on your self-confidence, on your ability to get advice when you need it, and so forth. And they were wonderful to me for a long, long time. I think what I've learned from them more than anything is the importance of paying back by doing the same thing for other young people. And again, without claiming that I've succeeded in doing so, I've sure tried to make it an important focus.
Well, that is wonderful. It's been great to hear a little bit about your role models, these people who've shaped you and really had a big impact on your career as you've gone through. And now I'm curious to hear a little bit more about your career. So can you take us, Greg, now all the way back to the beginning? Can you tell us about your earliest memories in your mind of being excited about science?
Well, I was always excited. I mean, I was interested in dinosaurs when I was a kid. People like Roy Chapman Andrews, the former director of the Smithsonian, who was the role model for Indiana Jones. I used to read his books when I was a kid. But I never thought I'd actually be doing science. I figured I'd be an academic, but I decided my real interests were in classical literature.
Really?
I went to college with the aim of studying classical literature. I like doing science, so I spent some time working as an assistant in Bill Horrock's chemistry lab, but that was largely to make money.
Gotcha.
But I ran into a couple of roadblocks in my career. One was that I'm lousy with languages, which is a bad thing if you want to be a classicist.
It is a problem, yes.
And the other was that I really couldn't make up my mind, if I wasn't going to be a classicist, whether I wanted to do anything at all. So I was playing for time, as it were. I was lucky enough to get a scholarship to go to England, and I decided what I would do was use that to spend 2 years doing classics over there and see if there was any way I could get past my language barrier. But while I was traveling to England, the guy I was going to work for died, and I arrived in England with nobody to work for.
Oh, wow.
So I called back to the United States, and Bob Langridge, who I'd worked with at Princeton, was British, knew the British system. And I said, Maurice Bauer is now deceased. I've got nobody here I can study under. What should I do? And he said, go over and see David Phillips and do molecular biophysics.
Wow.
So I said, okay.
Right.
Because other things, I was running out of coins to put in the payphone.
Gotcha.
And I walked across South Parks Road and went to David Phillips's office. And I said, hi, I'd like to work in your lab. And the great thing about the British, as you know, is they're very stoic, this whole stiff upper lip thing. Mm-hmm. And he looked up at me and said, who the hell are you?
Right.
And I said, I'm here on a full scholarship from the United States. I won't cost you anything. And he said, oh, okay.
In that case, completely different story.
In that case, it's fine. So, again, it was something I was only planning on doing for a year or two. But that time, I found I really had a taste for it. And I stuck with it. So that's how it all started. It's an amusing story. I hope at least people find it amusing, but that's not why I tell it. I tell it because I think there's a really important lesson here, which is I had no plan, no long-term life plan. I didn't have a clue what I was going to do. And that meant I was pretty open to more or less anything that came along. And I'm not saying that's the best way to live. What I am saying is if you don't have a plan when you're young, you shouldn't panic about it. It'll be okay.
Well, I think that is such an important lesson. I'm really, really glad you mentioned it. I think a lot of our listeners perhaps might be young scientists or young students who are trying to figure out what their next steps are, what their plan is. And it's really reassuring to hear very successful scientists like yourself say that they didn't have a plan going in, because I think you hear from a lot of people who, maybe started when they were 5 years old. They knew exactly they wanted to be a chemist. They wanted to be a physicist, and that's just what they did. But I think it's great to hear about people who kind of found their way into science through different paths. So can you tell us then what took you from that point of getting into a laboratory to where you are today? Are there some key moments that really brought you into your current field?
Oh, I think there were several. I probably thought more seriously about education than just about any other subject in my life. And one of the things I've always asked myself is, if we allow that my education seems to have been relatively successful in what it did for me, what was it about that mattered so much? And I think there were two things. One was that I never lost my love of learning. And the second was I never lost my confidence that I could learn. And so I think the crucial moments in my scientific life have been moments that have reinforced those 2 points, where I've discovered something that really excited me. And wow, I want to know more about this. And as a graduate student, that was how enzymes worked. We didn't know a lot about how enzymes worked. It was clear that if you understood their three-dimensional structure, you might learn how they worked. So, boy, that was something that I stuck with for 30 years. And similarly, when I first learned about neurodegenerative diseases, this just excited the heck out of me. And I wanted to learn more about it. And when I ran up against things we didn't know, I thought, okay, I'm confident that I can figure them out, maybe. So I think that that's been the critical moments in my life, have been when I've come up against things that were totally foreign to me, but really interesting and really exciting. And my love of learning and my belief that I was capable of learning enabled me to take the decisions to do those things.
Well, I think you hit on just two of the most important traits of scientists, this curiosity, this love of learning. But I really like this addition here, the confidence to know that you can figure something out. I mean, I think no matter what field you go into in science, there's still so much more to learn. Technology is advancing, so many new things coming down the pipeline that you're always going to have to be learning something, and you need that confidence to get through. So can you tell us a little bit more about one of these things that you're learning about now and a project you're working on that you are so excited about?
Yeah, I think that when we made the decision, by we I mean my scientific partner, Dagmar Ringo, who's professor at Brandeis, and me, we've run a joint operation for 35 years. When we made the decision to shift the bulk of our research into neurodegenerative diseases, we did two things that I think was very important. One was that we decided we would not work on anything unless we were pretty sure it was a valid target. And that immediately meant we weren't going to be doing most of what other people in the field were doing because we thought they weren't working on validated targets. And then the second decision we made was that we would work on more than one neurodegenerative disease because we thought that what we learned on one could carry over and help us understand the others. I think the moment we made those two decisions, it crafted our program in a way that was somewhat unusual at that time. And what I've been excited about is the fact that in the 12 years since we decided to do that, we've been able, with help of some great collaborators, to identify novel targets in Alzheimer's disease, Parkinson's disease, and Lou Gehrig's disease, and to develop potentially novel therapeutic approaches based on those targets for all three of those diseases. And now a lot of our work is focused on trying to see if we can get these things into the clinic so that we can finally see if they'll work in people.
Now, this is a phenomenal strategic approach. I really like it. So was there a strategic thinker among the pair, or did you both contribute to those two items?
We did what we always do when something important came along. We would sit in Dagmar's office. For some reason, she didn't want to sit in my office. She said something about needing a tetanus shot to go in. It is a bit messy, I must confess. We would sit in her office and just go back and forth, bouncing ideas off each other. You can't imagine how tremendous that is, to have someone in the next office who cares about the same things you care about. I couldn't put a price on it.
Absolutely. Well, wonderful to hear about the projects that you're working on in your lab today, and just thinking about the potential implications of this work, I think is so exciting. But we haven't yet chatted about the challenges, and I know there are challenges within this field itself and in science in general. So can you think back on your career, Greg, and tell us about one of these challenges that you faced, or a major failure that you had along the way that you were able to recover from?
I think that if you try to do almost any type of science, but especially if you're working at the cutting edge of your discipline, you're going to have far more failures than successes. And you have to learn to embrace failure and not to let it discourage you. We went through a period back in the early 80s when our lab probably didn't have a significant success on any of the projects we were working on. And we were working on about 6 of them for 5 years.
Oh, wow.
Longest dry spell I've ever had. We published a bunch of stuff, but it was just dribbling stuff out that wasn't all that exciting and important. To this day, I'm not sure quite what got us through that period. I think we got each other through, not just my colleague and myself, but the students and postdocs and all of us bucking each other up. But it was an extraordinary period of just nothing really panning out. I think when that happens, there's almost nothing else you can do except to roll up your sleeves and keep working and try not to be defeated. You're going to be discouraged, but that doesn't mean you're going to be defeated. There's a difference. Definitely.
So was there ever a point within that 5-year period where you considered maybe doing a major shift in your research or even leaving academia altogether?
Probably only about once a week.
Gotcha. On Fridays usually, or?
Yep.
That's fair. So what kept you on the path then? Was it just knowing your colleagues needed you, knowing your students needed you?
That certainly had a big impact, the students in particular, knowing that they depended on us. But I think equally important was the sense that this stuff matters. What you're doing when you're doing science is something that's important for the advancement of the human condition. Look, the reason you and I are having this conversation over Skype, instead of sitting in a tree wondering where our next banana is coming from, is because over hundreds of thousands of years of human history, people have dared to ask why. and to probe and explore the way the world works, to do experiments, to question authority, to try to find out what's going on. I think that's one of the most noble of human endeavors. Nobody ever said it was easy. Nobody ever said that you had a right to have it work out all the time or even most of the time. But the one thing you never had to ask yourself, no matter whether things were going well or badly, was whether what you were doing was important. And I think when you find something like that in your life, you hang on to it.
Definitely very well said. And I love this idea of really reflecting on during these low times when maybe you have 5 years of nothing working, just how far we've come and how 5 years in the scope of science is trivial. But just think about how far you can go in your lifetime and sort of in the lifetime of science, I think is so exciting and enough to, I think, keep anyone on the path. So wonderful to hear about how you got through this tough time. But next, I'd like to talk about successes, Greg. And I think successes in science are so exciting because they can be often very few and far between in some cases at some points in your career. So can you think back on some of your successes in science and share one with us today? This can be a big success or even one of these little victories along the way.
Well, I could share with you 150 successes. There wouldn't be a single experiment or a single publication in the list. It's taken me years to fully understand this. My successes, the success of me and my colleague, have been in the people we've trained. What matters most are the 150 or so graduate students, postdocs, and another plus undergraduates who've come through the lab and what's happened to them, the experience they've had while they were with us, and the life they've had after they've left. Those are the things that, when I look back, matter the most. Everything else is just the report card, right? What really matters are the people.
Absolutely.
I count as much of a success the 3 or 4 people who've gone through my lab and gone on to teach high school, and the several who've gone on to do other things like venture capital and so forth, as I do the ones who come out sort of clones of ourselves. To me, if a young person entrusts themselves to you, and at the end of that experience they've grown and developed as a human being and as a scientist, it's been a success. And if they've then gone on to have a happy, productive life, no matter what they're doing, then it's been a huge success. Those are the things I'm proudest of. I can talk to you about science, but it doesn't mean as much to me in retrospect as that.
Well, wonderful to hear about how you value your students and this mentorship relationship that you have with them. And I think it's so important for mentors to be supportive of the futures of their students, kind of whatever direction they decide to go. So I'm really, really glad that you said that. So can you tell us now how you're able to balance this role of mentorship with a lot of the research that you're doing? I know it takes up a lot of time to produce a graduate student from beginning all the way to end. How do you balance it?
Well, I think in my case, I balance it poorly, but I have the enormous advantage of not having to do it alone. The great thing about having this joint operation that Dagmar Ringe and I have run for so many years is that we have complementary strengths and we have reinforcing abilities. So I'm not necessarily good at dealing with some aspects of the personal situation in the lab, but she's much better than I am. And there are other things where I'm better at it than she is. And by dividing the labor in ways that matter like that, we can do a much better job than either of us could do individually.
Well, I think that is a really great approach. And I think it is important for a lot of young scientists and young people in general to have a variety of different mentors who all have different strengths or different things that they can teach them. So great approach there. And we chatted a little bit about what you like to do when you're not doing science in the beginning. And you mentioned you're a fan of writing. And I love writing. And I also love reading myself. So I love getting book recommendations from the guests we have on our program. And we mentioned a book where all of your contributions to the column on science and society are included within. But can you recommend for us, Greg, another book that you have really enjoyed and think we should check out?
Absolutely. I don't typically read science books for recreation. I like to read history, biography, and so forth. But there are 2 relatively recent science books that I can recommend with unqualified enthusiasm. One is The Emperor of All Maladies, which is sort of a history of cancer, and it's wonderfully written and quite engagingly told. And the other is Bill Bryson's book, A Short History of Practically Everything. And that's basically a sweeping survey of science. Bryson is a marvelous writer, but he also gets the science right, and it's tremendously enjoyable reading. In the non-science area, again, as a Bill Bryson fan, he has a new book called Summer of 27, in which he basically looks at the United States in 1927 and opens up an understanding of everything ranging from sports to culture to politics to economics, and it's fun, it's fascinating. I, of course, wasn't alive then, but my parents sure were. And I haven't been able to put it down since I started reading.
Well, excellent recommendations. I'll put those out there on our website for our listeners to find. And I myself, when I'm looking for a leisure book, you're right, I'm often not grabbing a science book. I'm looking for something in the literature, something, maybe a classic that I can kind of check out and enjoy. So great recommendations. Now, we mentioned your writing. So can you tell us a little bit more about that? How did you start writing on this topic of science and society?
Well, I've always been interested in fiction and biography and so forth, in terms of reading it. But when I decided I wanted to try my hand at writing things, I was driven almost by temperament to the essay form. The essay, as you probably know, was invented by Montaigne. And there are people who've just done such a marvelous job within the space of that short window of words in doing everything from entertaining you to really making you think. Most people know about George Orwell's novels, 1984 and Animal Farm, but in fact, he's one of the best essayists in history. Once you sit down and read his essays, they're marvels. So being drawn to that form, I then needed to sort of think about what I could write about, and the old advice about writing about what you know is good. So I started out writing essays about science and scientists and their life and society. I found out that people seem to enjoy reading them. I don't take a lot of things totally seriously, so tried to inject a little bit of frivolity into the essays, and people seem to like that. And that gave me the freedom eventually to branch out and write about pretty much anything I felt like.
Very, very cool. So is this something you're continuing to do? Are you continuing to communicate science and write about these issues in science and society?
I am, and I still am mostly in the essay format. I like writing essays. They're short.
Gotcha. Well, I know a lot of scientists are getting into blogging. Is this something that you've ever considered?
Yeah, I'm supposed to actually be setting up a blog for my Alzheimer's Institute where I offer thoughts about neurodegenerative disease and neurodegenerative disease research, and maybe I'll take that a little bit farther afield than that as well. And I'm hoping that'll happen actually next month. I'm approaching it with some trepidation. I don't want to do a blog the way a lot of other people do. I really do like the essay form, which is a little longer and requires a bit more preparation and thought. And I'm hoping maybe I can get away with sort of disguising essays as blogs.
Well, we won't tell anyone, right, listeners? Well, that is something for us definitely to be looking forward to in the next month or so, Greg. And we've chatted a little bit throughout our conversation about some of the opportunities that you've had to travel for these different positions that you've worked in. And I think travel is one of the fantastic aspects of careers in science, being able to work with different people internationally and also share your research across the globe. So can you tell us today about your favorite place that you've traveled for science?
Oh, that's easy, Australia. I love Australia. I've been there more than a dozen times. I never had a bad time there. It's a fabulous country. People are warm, friendly, down to earth. I think it's a bit like the United States probably was 40 or 50 years ago, which is a little hard for me to remember in a cultural sense. But geographically, if you look at the landscape, you look at the ecology, it's just an extraordinary place. Mind you, nearly all of the flora and fauna are venomous in one form or another.
Right. You have to be on your toes.
A country that's constantly trying to kill you, but if you take care of yourself, it's fabulous. I'd go back there in a heartbeat anytime I'm asked.
Wonderful. I've been to Australia myself as well and really enjoyed it. But can you tell us what brought you to Australia originally?
So what brought me to Australia originally was a request by someone I knew who happened to be Australian to participate in reviewing one of their institutions scientifically. And I went there and fell in love with the place.
Absolutely. Well, I know the highlight of my trip was holding a koala. Did you also have this opportunity?
Yeah, they're actually not that terribly warm and friendly creatures.
This is true, but it was still very enjoyable.
Oh, yeah.
And they tell you when you're holding them not to rock or shake them because they will try and climb you like climbing a tree when they get distressed. So
that's right. And they have pretty good climbing claws.
Yes, they do. Well, Gregory, wonderful to hear about your experience in Australia and just some of the great places that people get to travel for science. Like I said, is one of the wonderful aspects of the career that I think a lot of people aren't aware of. So we love to bring it up on our program. And I think one of the other aspects of the careers is this sense of collaboration, these people that you get to work with. And we've touched on this a couple times in our conversation, but I think every lab that you work with, every group of people that you work with has their own personalities. And I think they're often very contrary to this stereotypic view of scientists that a lot of people have out there today. So can you share with us a little bit of your take on the human side of science? Maybe some of these traditions or fond or funny memories that you have about being a scientist.
Oh, sure. I mean, you're quite right, of course. The great thing about being a scientist is that you're a citizen of the world, that you know people on every continent, and that you can go to almost any country and find someone you can talk to in a fairly intimate way about things that interest both of you. And that's been one of the joys of my life. I think for me, the experience of traveling to other countries and interacting with scientists there, especially collaborating with them, has been tremendous in terms of showing me that there's more than one way to work. You look at the Europeans, they work basically 9 to 5. Now, you'd think they'd never get anything done, but that's not true, because while they're in the lab, that's what they do. They work.
Right. They're productive from 9 to 5.
We take coffee breaks, we hang around the water cooler, we go sit down in the lunchroom for an hour and a half. It's just a totally different style. And it made me realize when I first encountered it that there are lots of ways to get work done, and that you don't necessarily have to impose a particular style on your group or on your people. And I think that sort of broadening happens a lot when you work with others. But also, if you choose your collaborators the way you choose your friends, I think that's really important, then in many cases they'll become your friends. And that enriches your life in more ways than I can count.
Absolutely. Well, great points there. I mean, I think this idea that there are lots of different ways to work really didn't hit home for me until I went to graduate school. And there were people who kind of rolled in at 11 o'clock but stayed there until 8 p.m. or people who were there at 6 in the morning but left by 2 or 3 p.m. So I think everyone has a different schedule. Everyone has different times where they can be productive. And I think different laboratories, too, often have their own sort of norm for when people are there, when people aren't there, and how people are working when they're there. So definitely a great point to bring up. And I think this idea of collaboration is so important. So can you tell us a little bit about how these opportunities for collaboration and maybe particularly international collaboration have sprung up for you?
Well, a lot of them happened at meetings. I went to a lot of scientific meetings when I was younger, partly to get my own work out in the public eye, but partly to meet scientists from other countries. And I eventually realized I didn't need to do that people are generally quite open to collaboration. And now I just do it frequently by telephone or by email. I'll see something that excites me and I'll just shoot somebody a message and say, I've got an idea for something we might do together. Are you interested? I'd say 90% of the time the answer is always yes, let's talk.
Definitely. Well, I think so important to think about science as a collaborative endeavor, not an individual pursuit. So great points that you've brought up. And we've chatted a little bit about your research throughout this interview, but one of the questions that I am a big fan of asking relates to some of the unanswered questions in your mind. So Greg, can you think about all of the restrictions that you face in science, things like funding, staff, technology, feasibility? If we took all of those out of the picture, if you could do anything, what is the question that you would want to answer?
In a sense, I'd go back to my structural biology roots a little, but I would ask a big structural question. What's it really like inside the cell? We think we sort of know, but do we really? I mean, when I was growing up, we thought of cells as just bags of proteins, of nucleic acids. Now we understand that there's a level of organization in there we couldn't have imagined. But I bet the level of organization is greater than we're imagining still. We know that the density of material inside a living cell is comparable to that of people in Times Square on New Year's Eve.
Wow.
By the way, I'm talking to you from about 40 blocks from Times Square. And this means that in order for living cells to function, there must be degrees of organization, traffic control, location specification that we're just beginning to have a sense of. I'd like to be able to peer inside a living cell while it's working and watch at the molecular level things actually happening. That's what I would do more than anything.
We just need a machine to shrink you up, pop you right inside a cell, we'll be good to go.
There are science fiction movies about that, but they don't end well.
Gotcha. We'll have to make a happier ending there. So Greg, if you had to think about, is there maybe one or a few things that you see immediately that are standing in your way from really being able to get a glimpse or an understanding of the inner workings of the cell?
Oh, I think the same thing that's standing in the way of half a dozen, maybe half 100 important things in science. The extraordinary risk aversion of the funding agencies. Mind you, one says that with some care. I don't like people who talk about the funding agencies as though they were monolithic, evil machines that we can't control. People say NIH has become very conservative. Well, I hate to say it, but we're NIH. The people who review grants are us.
Right.
The people who review papers are us. So the fault, as Shakespeare wrote, is not in our stars, it's in ourselves. And I think we need to understand that. But having said that, I think that we become extraordinarily risk-averse in terms of what we pay people to do right now, in terms of public funding of science. And this troubles me enormously.
Certainly. I think it is one of the issues of the times and sort of human nature almost to tighten your pocketbook in times when funding is scarce. But it is a problem that is impeding some of the maybe riskier, maybe less well-established science from moving forward. So great points there. And we've chatted throughout our conversation about some of these people who've had a big impact on you and your career. And I know as scientists, you get a lot of fantastic advice as you're going through the process at all stages. So can you share with us, Greg, what you would consider the best piece of advice you've ever received?
My first faculty job was at Wayne State School of Medicine in Detroit, which was, at that time, to be kind, a second-rate institution. It had, however, first-rate human beings. So I understood eventually that one of the most important things about where you start your career is that it be a great place to start in terms of emotional support, not just intellectual support. And that was a great supportive environment. If you could see my office now, you'd see the only picture of people I have in my office is faculty picture from Wayne State University. But the point is, when I got there, because it was a second-rate institution in those days, I spent a good deal of my first year there trying to figure out how to get out.
Gotcha.
Sent applications off for jobs to other universities, started worrying about whether or not I would ever leave, and so forth. And there was a senior faculty member there named Richard Hudson, and he actually noticed this. And he took me aside and said, Greg, I think you've got a lot of ability, but if you spend all your time trying to figure out how to get out of here, you never will. Do the best work that you possibly can. And if it's meant to be that you should be elsewhere, the opportunity will present itself because you have done great work. I think in the course of your life, you hear all kinds of advice and you don't pay attention to it when you should. But there was something about that moment that I was actually ready to listen. And I took that advice to heart. And 5 years later, I got an offer to move to MIT. And the funny thing was, when I got that offer, I almost didn't move.
Really?
Yeah, because I had come to love the people at Wayne State so much. So there's a richness to it. If I'd spent all my time trying to figure out how to get out of there, I wouldn't have enjoyed being there. Once I decided to forget about that and just focus on the work, A, I got the opportunity to leave, and B, I came to love where I was much more than I would have otherwise. That was a lesson I've never forgotten.
Well, what a great lesson. What a great story really built around it. And I think really important for scientists to think about this idea of the grass is always greener on the other side. And, you don't really appreciate what you have until you're ready to leave. And I think that is a powerful story for a lot of scientists who might be concerned about their career prospects and maybe accepting positions that are not their ultimate goal. But you have to think about being the best scientist you can, being the best person you can, making connections where you are. And you're absolutely right. I think the right opportunity will present itself if you're able to do that. So fantastic story. Now, can you share with us, Greg, one last piece of advice or perhaps one last piece of inspiration for myself and our listeners today?
I don't consider myself particularly wise, but I've learned something over the years that I think is very important to keep in mind, and that is that you are what you do. I think that it's too easy to get caught up in trying to understand people's motivation. It's too easy to get caught up in your own motivations sometimes. In the end, the kind of person you are is really indicated by what you do, by how you conduct yourself. And I think if you pay attention to that, both in terms of the work that you do and in terms of the way you treat other people, you have the best chance of ending up the way you want to be. God knows it's not perfect. I mean, we could have a podcast 10 times longer than this one in which I talk about all the mistakes I've made and how much I regret them. But the fact is, in the end, I have no particular insight into what's in somebody's heart or soul. I don't know whether their intentions are good or bad. But what I do know is if they do good things, then at some level they're a good person. And if they don't do good things, if they do things that are not good, then intentions be damned, they're not behaving like a good person. And I think keeping that in mind, the way you conduct yourself and the way you evaluate others, probably not a bad thing to do.
Very insightful, Greg. And I think this is definitely something to keep in mind. You said you choose your collaborators like you choose your friends. Kind of keep this in mind. Are you going to be collaborating? Are you going to be working with good people? And I think surrounding yourself with good people really makes life and science so much easier and happier. So
if you asked me what the best moment of my life was, I wouldn't know how to answer that question. But if you had asked me what the best moment of my scientific life was, and it'd be pretty high on the general list, it was the day Dagmar Ringe decided that she wanted to work with me, that we set up our joint operation and collaboration. To find a colleague and a friend like that will enrich your life in a way that you can't possibly begin to describe in words.
Absolutely. I think having a partner as you're pursuing these questions that you're excited about in science and facing the challenges together, I think is a wonderful way to go through science. Not being alone in this exciting, challenging roller coaster of an endeavor, I think is such a great way to do it. So Greg, thank you so much for sharing your insights and your stories with us today.
My pleasure. Thanks for having me.
Absolutely. Now, can you tell our listeners how they can reach out to get more information about you and your research?
Sure. I don't do social media. I'm probably, if anything, anti-social when it comes to social media.
That's allowed.
I like to think I'm pretty responsive to email. My Cornell Medical School email address is gpetsko, all one word, @med.cornell.edu. And I am happy to respond to emails, although I'm also somewhat erratic in terms of the frequency with which I get to them. I'm also happy to take phone calls or have people write me real honest-to-God letters. I haven't gotten one of those in months, and all of that is fun.
Well, fantastic, Greg. We appreciate it. And thank you to our listeners out there for joining us today as well. We'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.